And first of all, I appreciate it very much for being invited here to this to give a talk in this JIRCAS seminar, international seminar, in connection with the United Nations International Year of Millets 2023. I would like to address my sincere thanks to the organizers. And also, I'm very happy to discuss with you, and particularly with Dr. Jacqueline Darroch-Hugh, ICRISA DG, and Dr. Sitara Satyawati, Director of IIMR. Maybe my talk is not so systematic as Dr. Hugh, but my talk is titled Reappraisal of Millets, with a long history of cultivation, may inspire sustainable survival of humankind. It is my opinion that we should be more serious about agriculture in order to survive sustainably in the VUCA era, with the explosive population growth, global heating, pandemic, food security related with refugees, war and conflict, political confusion, and so on. Food and Agriculture Organization, FAO, reported that 90 or 95% of human food energy needs come from just 30 crops. Almost half of our daily calories come from rice, wheat, and meats. We have improved major crops, often toward monoculture of a few varieties on a large scale, relying on the fossil energy and chemicals. Please pay close attention to so-called minor crops, such as millets, and reappraise them, which have been our partners since the time of early agriculture. They have lower productivity than major crops at present, but a wide diversity and resilience against harsh conditions. The Japanese word zakkoku, which does not always mean millets. Cultivated crops are conceptually grouped into 4 categories traditionally in Japan: shukoku, zakkoku, shukukoku, and gikoku. Sorry in Japanese. The zakkoku can be used for various crops because it's often sold as complement set of shukoku. It sometimes includes leguminous crops and or cereals in Japan. Maize is a major crop in the world, but sometimes included in zakkoku in Japan. When I talk about foxtail millet, my material, foxtail millet, with an Indian scientist, it took us a while to reach a common ground because the word millet meant pearl millet for him. He said, "Oh, you are talking about small millets." Time to time, place to place, the word millets may differ. Today, I believe we are already on the common ground. Oh. Enter. Well-known millets in Japan are foxtail millet. Maybe. Can we use, no, okay. Foxtail millet, common millet, Japanese barnyard millet, Job's tears, finger millet, sorghum, as shown in the slides. Sorghum is usually taken as an animal feed and fodder in Japan rather than a cereal crop. Millets, especially foxtail millet, is one of the first crops born from the body of goddess in Japanese myth. Maybe many Japanese knows well. Rice and foxtail millet are described as the 2 cereals used for the celebration by the emperor for the good harvest and for a fruitful new year, as Niname Sai, Niname no Matsuri. Millets are used for rituals not only in ancient times, but until now. Millets production and economy are often small and localized compared with major cereals. Modern breeding practices are limited in certain millet species. Local land races are still grown, but have not been intensively explored. Some species are not studied or evaluated. Their cultivation is actually decreasing, but still maintained, since they have their own cultural importance among local people. Most of them are summer-grown C4 plants, since they were domesticated in tropical savanna or temperate semi-arid zones. Origins and crop evolution of millets are not always clear. They were domesticated in Africa, South Asia, and Eurasia, including China, and some exceptions in South and Central East Americas too. Oh, it's a lagi finger millet in South India. Foxtail millet in Japan. Common millet in Japan also. It's corn in South India. It's Raishan in Meghalaya in Northeast India. Kodo millet. There are so many sorghum, foxtail millet, and palm millet, and finger millet in the same field in India. It's samai, so little millet, and what was the name? Kolari, very minor millets, and Job's tears. There is no evidence that millets were cultivated in China before 11,000 years ago, but people did gather grains from their wild relatives in Yellow River Basin. Millets cultivation dates back more than 7,000 years ago. Remains of foxtail millet noodle were excavated, unearthed from a site dating back about 4,000 years ago. It was found in China. So the introduction of agriculture to Japan is a new event compared to China. Cultivation of foxtail millet and rice in northern Kyushu now dates back roughly 2,800 years ago. So traditionally, Japan has a strong sense of rice supremacy, especially regarding white rice. Rice has been a king staple food cereal crop for a long time. Rice harvest was collected as tax. However, rice production was not sufficient or stabilized from ancient times. Millets were subsidiary crops together with wheat, barley, yam, taro, and others. Cultivation of millets were repeatedly recommended from ancient times against famine. Millets have been our partners since the time of early agriculture. So cultivation of millets decreased drastically in Japan during the last century and have not been recorded in government statistics since 1975. Although they have lost their economic importance, they are still maintained in a small scale because they have a unique cultural significance among the local people. Recently, millets are attracted attention due to their nutritious value and low allergenicity. Moreover, new generations, freed from the idea that millets are poor man's food materials, recognize the value of millets. Civil society organizations and municipalities are exploring new ways to promote millets production and new uses. These have potential for improvement as they have been often neglected and underutilized without experiencing green evolution. Then I would like to talk genetic diversity from our experiences, especially in foxtail millet. Among various millets, foxtail millet is characterized by morphological variation and wide distribution as a global millet. We found hybrid sterility between local varieties. We call local varieties as land races. For example, when a Japanese landrace from Japan is cross-pollinated by another Japanese landrace, we usually obtain fully fertile pollen and seed set in the hybrid. On the other hand, lower one, when a Japanese landrace is crossed by a landrace from Taiwan, we often get sterile pollen and a few seed set in the hybrid. So this hybrid sterility, actually semi-sterility, suggests a differentiation between the parental land races. Based on the hybrid sterility, we classified land races collected from various parts of Eurasia in at least seven types. Types A, B, C, D, AC, BC, and X. Those types have their own geographical distributions. Type A is mainly distributed in Asia. Type B is restricted in Taiwan and adjacent Okinawa Prefecture of Japan. Type C is widely found from Europe to South Asia. Type D is limited in the Lanshu Island of Taiwan and the Bataan Island of the Philippines, and isolatedly distributed in northern Pakistan. Type AC is mainly Central Asia, including Afghanistan, and type BC is mainly in South Asia. Distinct landrace groups with specific geographical distribution are also supported by DNA polymorphism. These studies tell us a picture of genetic differentiation through crop evolution along with the history of agriculture. Based on this diversity analysis, 88 accessions of foxtail millet and 6 of wild ancestors were selected as mini core collection by narrow gene bank. In the foxtail millet, there are 3 starch types: non-waxy, in Japanese, uruchi, waxy, mochi, and intermediate, chukan. Okay? Waxy means glutinous. Amylose content controls this character. Non-waxy type, so left upside. Non-waxy type is found through Eurasia. Waxy type is grown in East and Southeast Asia, and intermediate type is distributed in Southeast Asia. A gene, GBSS1, is responsible for biosynthesis of amylose. We found that GBSS1 gene has evolved by complex inserted transposable element shown in left low hand. Evolutionary pathways of this single gene clearly visualize crop evolution and geographical distribution of this millet. Through the cultivation, our ancestors selected different mutations for our food preference, okay, without looking at the genetic makeup. This kind of basic information can be utilized not only for improving foxtail millet, but other crops. Recently, one of my students conducted a very simple screening for salt tolerance in foxtail millet using the aforementioned mini-core collection. It is interesting to note that in some soil areas affected by salt-tolerant landraces have evolved. The mini-core collection is a small number of accessions selected to represent as much as possible the entire genetic diversity of the mother collection. For such studies, it is very important to collect genetic resources. Even such a mini-core collection can be used to search for and study useful traits. And their genetic mechanisms can be now studied and exploited using the modern genomic tools. Fukunaga and his group have reviewed the genetic control of heading date, plant color, panicle shape, and so on in the foxtail millet. Similar studies are done by many scientists in the world. Those studies are sometimes called crop evolution studies. They are not only basic or retrospective studies. Such studies can be compared with other crop plants and are adaptable to the improvement of other crop species too. In other words, the differentiation of land races, environmentally adaptive characters, and traits related to human preference provide hints and inspirations for the future use of these millets, as well as the improvement of other crops. Such studies cannot be done without the worldwide collection. Genetic resources are indispensable. Narrow gene bank holds 1,687 accessions of foxtail millet from Japan, China, India, Pakistan, Korea, Myanmar, Nepal, and so on. Are they enough? Are they deeply researched? To shed light on underutilized and neglected plants like millets, we need to collaboratively explore and conserve as much of the existing diversity as possible and study and evaluate their diversity cooperatively. As Dr. Hugh said, I agree, Dr. Hugh, for future cooperation, the importance of future cooperation she suggested. Thank you very much for your kind attention.
Ladies and gentlemen, distinguished participant and guest. Today, we, Yoshihashi and Subbarao over there, share the presentation of our activities on sorghum and millets. The first, I'd like to explain the BNI technology, which addresses both food and environment security. In fact, our civilization is highly dependent on food production system, and we have several critical concepts in history. The first one is a concept of the fertilizer introduced by the Dr. Levy, as shown here, and then industrial nitrogen fixation, the Haber-Bosch process, was developed to realize this concept. So the human beings start to be disconnected from the environment. So with the energy we can produce any amount of the nitrogen fertilizer. At present, modern agriculture relies heavily on the concept of the green evolution, the concept of the high-yielding varieties and resource input introduced by the Dr. Borlaug. we achieved this only 50 years ago. So the farming is almost disappeared from the 50 years ago. We believe that it provided enough food to enjoy other activities because we don't need to search food. We have some activities other than the agriculture. However, our planet Earth is a finite system, which means it has limits. As shown here, the industrial fixed nitrogen is already pushing the limits of the Earth threshold, here nitrogen cycle, due to our food production. The reactive nitrogen from the nitrogen cycle with the toxic nitrate contamination of the groundwater and intense greenhouse gas emission as a nitrous oxide, which is 298 times more potent than the carbon dioxide. However, the food security is our primary concern. We still need to rely on this green revolution technology. Currently, over 50% of nitrogen fertilizer is not used by the crop in agriculture. Because of the nature of the soil, periodic application of the ammonium induces faster nitrification by bacteria, which converts the soil fixable ammonium to the easy rich nitrate. We are working the biological nitrification inhibition, we call the BNI. The crop produces nitrification inhibitors to retain the ammonium in the soil, and the crop uses ammonium intensively. And as a result, the nitrogen use efficiency and environmental impact of agriculture have been improved. We aim to improve the nitrogen cycle through the deployment of this BNI technology to minimize both nitrogen fertilizer use and pollution from the excess nitrogen application by agriculture. This BNI is phenomenon which was found in 1983 on the tropical pasture, Brachiaria humidicola, and we JIRCAS initiated this research in 1995. Then we reported the evidence of this BNI in the Brachiaria humidicola in 2009 and proceed to explore other crops for the BNI capacity. Actually, it takes a long time. Since we targeted the food crops further, we initiated upland crops such as sorghum, wheat, and maize. In 2015, we initiated research activities on wheat and finally found wild wheat, Limas racemosus, has a BNI capacity, and its BNI capacity was successfully introduced into the modern high-yielding wheat. And we confirmed and achieved the BNI-enabled wheat development as a proof of concept of this BNI technology in Japan in 2020. It is a quite long journey. It has been a quite long journey for us to obtain this prototype of the BNI technology and BNI-enabled wheat. And the story is going on in other crops like in sorghum. And then today we will explain about the millets. Besides, the JIRCAS is formulated the BNI International Consortium with a research institute from all over the world, together with the CGIAR centers like ICRISAT. We had organized the fourth biennial meeting in November last year, and then we will organize next year in November 2024 in Tsukuba in Japan. And JIRCAS is conducting its own research project on the BNI research, which includes additional funding. And we are looking at the wheat and maize, which receives higher nitrogen fertilizer in the field, and sorghum and brachiaria pasture, which receive less nitrogen fertilizer, and expected to add millets. It should be the finger millets today, Dr. Subbarao will present. and which targeted lower nitrogen application to develop the planet-friendly genetic mitigation that balances food and environmental security. And today's topic is our activities on the sorghum and finger millet as a typical low nitrogen fertilizer receiving crop. So floor will be changed to the Dr. Subbarao.
Good morning to everybody. And thank you, YOSHIHASHI-san, for giving the overview of the BNI research in JIRCAS and what we are doing from the last 20 years. As my colleague has mentioned that last 6 decades of green revolution, which relied mostly on application of factory-produced nitrogen fertilizers to agricultural systems that has transformed the agricultural production and brought the food security to all of us. But it also has brought many negative impacts in our ecosystem and also the environment and the climate. You all know that the nitrogen fertilizer applications have grown immensely since the last 6 decades, starting from around less than 2 million metric tons to nearly 150 million metric tons what we are currently applying. Several fold, probably more than 40-fold increase in nitrogen fertilizer applications from the last 6 decades. But it has resulted in only about a 4-fold increase in global food production. So we are applying more and more nitrogen fertilizer now to just maintain or just a marginal increase in food production. And that is causing a lot of problems, as I mentioned before. When we apply nitrogen fertilizers to the soil, it is aimed at the crop rooting systems. The crop root systems should be able to utilize this nitrogen fertilizer and take it up and convert it into plant protein. That is the aim. But what is happening is the nitrogen fertilizer, when you apply to the soil, it has to go through the plant microbial interface before it can reach the plant root systems. Since this last 6 decades of green revolution has brought enormous changes in the soil physical and biological properties, particularly a section of soil bacteria, which we call nitrifiers, they have become out of control now. They compete for nitrogen fertilizer along with the roots. So the competition between plant root systems and this soil microbial component is so intense now that this nitrifier community in the soil bacterial community, part of the soil bacterial community, nearly takes up more than 98% of the nitrogen fertilizer what we are applying. So the plant roots have a very little chance to take it up, this nitrogen fertilizer what we are applying. What's happening is the crops are left with the microbial byproducts, not the nitrogen fertilizer what we are applying. Most of the nitrogen problems what we see in agriculture are not coming from the nitrogen fertilizers directly. It is coming from the microbial byproducts of this nitrogen fertilizer. Because we are unable to control this soil bacteria, that's the reason it is using up all this nitrogen fertilizer and changing into these harmful nitrogen byproducts such as nitrate, nitrous oxide, nitrous monoxide. And these harmful nitrogen byproducts like nitrate, which can be used to some extent by crops, but they're not very efficient because they can't bind to the soil. That's the reason they're leaking out of the agricultural systems and causing all these problems and also causing low nitrogen use efficiency. So the only way we can control this nitrogen problem is infusion of large scale infusion of the antibiotics from the plant root systems that can really control this nitrifier activity in the soil. That is what is called the biological nitrification inhibition. This is a concept we have introduced in 2009 and proved that the root system, plant root systems produce these specific antibiotics that can control this nitrifying community. That's how the nitrogen, the fertilizer can be protected from this soil bacteria and make available for the crops. Since this is a biological function, this can be genetically modified. This can be bred into crops. This can that's how the next generation agricultural systems can control this the bacterial activity in the soil, so that the nitrogen fertilizer can be available for the crops, which is intended, which is the intended purpose. We have developed an assay system in JIRCAS in the beginning of our BNI research that can detect and quantify the amount of nitrification inhibitors. This is specific antibiotics coming from plant root systems, and so and quantify the amount of this inhibitory activity coming from the plant root systems. Just to give you an example, this is what you see is the Brachiara. It's a pasture grass where this function was detected in the beginning. And a plant like that can produce up to 200 to 300 units of inhibitory activity per day. And a gram of soil requires about 5 units of inhibitory activity to reduce the nitrate production to 50% from what from the current levels. And most plant root systems produce 2 categories of the inhibitors, the antibiotics, the hydrophobic type of antibiotics, the hydrophilic type of antibiotics. And the relative contribution and of these hydrophobic versus hydrophilic vary from crop to crop. For instance, in finger millet, nearly 70% of the total antibiotic production is coming from the hydrophobic type. Only 30% is coming from the hydrophilic type. In wheat, it's 95% is coming from the hydrophilic category. Only less than 5% is coming from the hydrophobic category. In sorghum, it's 60% of the total inhibitor production is coming from the hydrophobic, and only 40% coming from the hydrophilic. In maize, it's 40% is coming from the hydrophobic, and 60% is coming from the hydrophilic. Sorry, 60% is coming from the hydrophobic, and 40% is coming from the hydrophilic category. So the relative contribution of the hydrophobic versus hydrophilic would change from crop to crop. So the functional value of hydrophobic versus hydrophilic also changes. The hydrophobic ones are mostly confined in the rhizosphere, so they are not linked to the presence of ammonia in the rhizosphere. So which means under low nitrogen input conditions, hydrophobic activity is more important. Their expression is mostly under low nitrogen environment conditions. They're relevant for low nitrogen environment conditions and are also likely to be associated with soil organic matter mineralization and extraction of nitrogen from the low nitrogen input conditions. Whereas the hydrophilic BNI activity is mostly associated with the high nitrogen input conditions. And their release is also linked with the presence of ammonia in the rhizosphere. And they are expressed mostly under high nitrogen environments, and they're not confined to the rhizosphere. So the relative contribution of these hydrophobic versus hydrophilic vary from crop to crop, and depending on the production system, they're evolved. And this is the release of hydrophilic BNI activity, which is linked with the presence of ammonium. And the presence of ammonium, the uptake and the assimilation are some of the factors that drive the release and production of these hydrophilic BNAs from the plant root systems. So that's the reason the plant root systems, when they sense ammonia, which is coming from the nitrogen fertilizer applications, then only this part of the inhibitory activity is coming from the root system. So it's a highly regulated plant function, the hydrophilic part of the BNI function. And also the plants produce a variety of these inhibiting compounds or the antibiotic compounds. So our group has isolated several compounds from the tropical pasture grasses like Brachiara and sorghum, and recently maize. So for sorghum, the main component of this inhibitor is sorgoleone. So it's the major component of hydrophobic BNI activity. And since this accounts for almost 90% 90% of the total hydrophobic component of the BNI activity. All our work on sorghum is focused on improving the sorghum production from the plant root systems. So we started this work with ICRISAT a decade ago, and we have now identified a number of sorghum germplasm lines that has almost more than tenfold higher sorghum production than some of the high-yielding sorghum varieties, which gives us a possibility that we can improve this plant trait and develop a high sorghum producing sorghum varieties in the future, which I will talk a little bit later. And this is the nitrosomonas, the bacteria, which is responsible for consuming most of the fertilizer nitrogen in our farmlands. So it uses two enzymatic pathways, AMO and HAO pathways, to oxidize ammonium, basically to consume ammonium into as an energy source for this bacteria. Most of the synthetic nitrification inhibitors that are developed only target the AMO pathway, but whereas the nitrification inhibitors are the antibacterial compounds coming from the plant root systems, which is responsible for the BNA function. They target both AMO, HAO, and also the electron transfer pathways. So they have a multi-mode of inhibitory action. So the inhibitors that are produced from the plant root systems have a multi-mode of inhibitory function. That's the reason it is likely to be more stable in the long run than the synthetically produced nitrification inhibitors to control this bacteria in the soil. So as I mentioned that our work on sorghum started nearly about a decade ago with a collaborative partnership with ICRISAT. This is one of the field trials what we have conducted. We have been conducting field trials from the last several years. So this is one of the field trials, the BNI field trials in ICRISAT. And as I mentioned, nearly 60% of the BNI, the inhibitor production in the sorghum roots coming from the hydrophobic. That is, most of it is coming from the sorghum, and only the remaining 40% is coming from the hydrophilic, the BNI production. And this is how it looks, the root exudate coming from the sorghum roots over there. After 24-hour collection, that is how the root exudate looks, which contains a lot of this inhibitory activity. And this is the range of variation what we see in terms of sorghum production from the sorghum roots from a mini core sorghum collection of about 200 lines. So you can see on your right side the PVK 801-296B. Those are the two elite sorghum lines, which are that reflects most of the high-yielding sorghum varieties coming from the sorghum breeding program, based on those two lines. So the sorghum production capacity, what you see in most of the high-yielding current modern sorghum varieties, is only about 1/10 of what could be improved based on some of the high sorghum producing lands what we have identified coming from West Africa. And currently, we are developing a number of mapping populations based on some of the high and low sorghum producing lands identified in the mini core. And these new mapping populations, which we have started developing about nearly 5 years ago with ICRISAT partnership, now they have reached a stage where we can now take up the phenotyping of these mapping populations, and which we would be starting from next year. And this requires a lot of support to take up this activity in order to identify the genetic markers that are linked with the sorghum production, which would be helpful in developing the high sorghum producing sorghum varieties in the future. And our work on finger millet just started. This is through partnership from IIMR in India. As I mentioned, that the hydrophobic BNA capacity is a major component of the BNA function in finger millet. Nearly 70% of the the inhibitory capacity is coming from the hydrophobic BNIs. Only less than 30, only about 30% is coming from the hydrophilic. So hydrophobic is much easier in terms of evaluation and probably the breeding for high BNI function can be easily taken up because it's a hydrophobic component, which is relatively easy to phenotype, scale up. And recently, we started isolating the major, the component responsible for the hydrophobic BNA activity in the finger millet. And we are pretty close to isolating one of the major components. Probably by the maybe in a year from now, we will be in a position to come to you with the main component, the chemical structure of this inhibitor coming from the finger millet. And also we have received nearly 250 mini-core collection of finger millet from IIMR through ICRSAT. And we will be taking up the phenotyping for the hydrophobic BNA activity of finger millet. But this depends on the additional funding available for JIRCAS to take up this activity. And by improving the BNI function in the plant root systems, we can shift the nitrogen uptake in our crop plants from the currently is nitrate-centered nitrogen nutrition. More than 95% of the nitrogen uptake is through the nitrate because of our inability to control the nitrite activity in the soils led to almost all the nitrogen what is available in our farmlands is nitrate based. So though we are applying the nitrogen fertilizers are mostly applied in ammonium form, but the crops are taking up only in nitrate form because it is all consumed by this bacterial activity, and we are left with all these harmful nitrogen byproducts. But if we improve the BNI function in plant root systems and develop the crop varieties that have a very high levels of this ability, we can shift the nitrogen uptake more and more towards ammonium. That is what is going to solve the nitrogen pollution problems from agriculture, and also that's what it's going to solve the nitrogen nitrous oxide and also all the greenhouse gas emissions coming from the agriculture. This is how we can solve by introducing the BNI function, by developing the BNI systems in our agriculture. Will have a major impact in terms of controlling the nitrogen pollution, in terms of reducing the GHG emissions. That includes nitrous oxide. This is what published recently in PNAS. And it's not only reduces the nitrogen pollution, but also improves the ammonium form of nitrogen availability. That would have a positive impact in terms of growth and productivity. If you improve the ammonium form in our farmlands, that has a very big stimulatory effect on crop growth. If they depend completely on nitrate based, the crop growth and productivity can be improved if we shift a little bit more towards ammonium based. It's a balanced form of nitrogen forms like ammonium and nitrate will have a very big impact in terms of improving the productivity. So that can also has a very big impact in increasing the agricultural productivity to meet the expected challenges in terms of overall improvement in agricultural productivity, which is about nearly 50% more than what is currently done by 2050 is needed. So this could be one of the key technologies that should be developed. And our work on BNI wheat, my colleague has mentioned, Dr. YOSHIHASHI, that we have developed a prototype of BNI wheat by transferring the key trait of that is regulating the BNI function in wheat from a wild wheat into the cultivated wheat. Now we have several elite wheat varieties carrying this chromosome segment controlling the BNI function in the root systems of cultivated wheat. So this work was awarded Reappraisal Prize by the National Academy of Sciences US in 2022. And I think our work on millets that is mostly focused on sorghum And also we have started recently working on pearl millet and the finger millet. So these three comprise almost the coming under the major millets. So almost 90% of the total millet production in the world coming from these three millets. I think our work would be expanded to these three millets in the near future. And we hope we will have the support to do that. Thank you.